Уважаемые коллеги, проходите, пожалуйста, в зал, и мы начинаем. Сейчас залы будут проходить в 3 залах.
Дорогие коллеги, будем начинать сессию через пару минут. Просьба занимать всем свои места. Пожалуйста.
Уважаемые коллеги, просьба...
Dear colleagues, please take your seats. We are beginning our session. Dear colleagues, to keep the time frame, because we have an agenda and we see that lots of people follow this plan. So if everybody has 15 minutes for your presentations, please don't use more time, because others also would like to speak. Diagnostics is always the beginning of any infection. This is what it begins with. And also, this is the final stage demonstrating that this is the infection we have been speaking about. Okay, and how fast we are in terms of diagnostics. This is the prerequisite for successful epidemiological measures and epidemiological forecasts. That is why I believe that we are going to have a very interesting session, and it is very significant within this event. I'm going to pass the floor to Christina Deschermeyer, Bernhard Nocht Institute for Tropical Medicine in Hamburg, Germany. And Christina is going to speak about COVID-19 diagnostics, problems and solutions. 20 minutes, please.
Yeah, first of all, I want to thank the organizers to give us the opportunity to talk here at this exciting meeting. So my name is Christina Deschermeyer, and I'm working as a research scientist and a lab group leader at the Bernhard Nocht Institute for Tropical Medicine in Hamburg in Germany. And I want to talk about SARS-CoV-2 serological diagnostics and the problems that we encountered there and the solutions that we have found for these problems. Just as a short introduction of our institute, so Bernhard Nocht Institute for Tropical Medicine is localized in Hamburg, Germany, directly at the side of the River Elbe, And we have two buildings. We have this old historical building here, built in 1914, and then we have a new building that harbors our new BSL-3 and BSL-4 laboratories. So our institute is focusing on tropical and emerging infectious diseases. So there we do research, teaching, training, counseling, and also ambulant patient care. And the institute serves as a national reference center for tropical pathogens in Germany and as a WHO collaborating center for arbovirus and hemorrhagic fever reference and research. I myself had the R&D department of the Diagnostics Development Laboratory at BNITM. And in this lab, we are focusing on the development, optimization, validation, and the production of serological assays. And thereby, we closely cooperate with specialized BNITM departments, being specialized in the respective pathogens, and also a lot of institutions in endemic regions with a strong focus also on Africa. In addition, we also evaluate commercially available diagnostic tests, and we provide ELISA training and teaching. So during my talk, I want to first focus a little bit on the applications of SARS-CoV-2 antibody binding assays, so assays that detect the presence of antibody recognizing viral proteins. And here we are mainly focusing on ELISA and immunofluorescence techniques. So, of course, for the acute diagnostics of acutely ill patients, the molecular diagnostic techniques like PCR and also antigen detections are much more important. But from time to time, also antibody diagnostics for individual patients is necessary. For example, for people wanting to travel to China, they have to provide IgM negative testing to be able to just enter the country. So this is also a service that is provided at our institute. And then a large area where SARS-CoV-2 antibody binding assays are needed are seroprevalence studies. Here our institute works very closely together with several of African countries, because especially in countries where testing capacities are low and not so many PCR tests can be performed, the case numbers are seriously underreported normally, and here seroprevalence studies can help really to estimate the disease burden, to estimate morbidity and mortality rates in these countries. During our work on the SARS-CoV-2 antibody binding assays, we encountered several challenges, and these are the ones I want to focus on in my talk here, and I want to summarize them up here, and then we go through it point by point. So one is that the assay sensitivity, or that the antibody response during reconvalescence from COVID-19 disease is waning over time, so antibody titers decline. So the assay sensitivity will also change over time, and this is very important in the context of the seroprevalence studies, because then it's important when the sampling is done respective to the waves of infection in that country. And as we experienced also different assays, also commercially available assays for antibody binding, have very different sensitivities, so it's always difficult to compare results generated with different assays. Another problem that we also encountered in our ELISA studies is that although the assay specificity often is very good in European samples, we recognize that when applied to serum panels from different world regions, for example, especially in Africa, we saw that a lot of the commercial available assays don't have enough sufficient specificity to really allow reliable seroprevalence studies. And then the last challenge we also encountered is when we saw that detection of breakthrough infections in vaccinees also has certain problems. First of all, before we come to the sensitivity topic, we at our institute developed our own in-house SARS-CoV-2 ELISA assay, and to validate this assay, we established also our own patient cohort. And how this looks like is described here. So what we did was that we recruited 35 probants from Germany with a confirmed SARS-CoV-2 infection. So all these patients had a PCR positive swab in between the March 2020 and March 2021. And all the patients had mild disease, so none of them had been in hospital. And we asked those patients to come back to our institute to give samples every 1 to 12 weeks. So in the beginning of the reconvalescent phase, they came a little bit more often. So that now we have for each of the probants between 1 and 13 post-infection longitudinal samples, so that we can follow the humoral immune response in these patients for a long time, and up to now we arrived here at a little bit over 12 months of time post-infection. A lot of our probants are healthcare workers, so a lot of them also received pretty early on in January 2020 already, January 2021, already a spike-based COVID-19 vaccine. And what you can see here is the trajectories for these 35 different patients, where we analyzed the serum samples with immunofluorescence. So there we had virus cells that had been infected with SARS-CoV-2, and we just quantified the amount of SARS-CoV-2 directed IgG antibodies, so not differentiating between different antigens. And as you can see here for the different patients, this is a little bit easier to see here in that stratified representation here. Most patients had the highest humoral immune response, the highest IgG immunofluorescence titer 1 to 2 months post onset of symptoms, and then over time the antibody response dropped, but still detectable levels of antibodies were detected around 12 months post onset of symptoms. When the patients received their vaccination, then the immunofluorescence titers raised very quickly, as you see here, to these dark filled dots here. These represent the titers post-vaccination. So the spike-based vaccines strongly boost the humoral immune response in these patients. Then we used those samples to validate our in-house ELISA assay. So our in-house ELISA assay is based on a patented technology where we have an immune complex binding molecule that we coat to the ELISA plates, and then we apply together with the patient serum a labeled antigen. In our case, this is the nucleoprotein-based antigen. Then immune complexes are formed, and these immune complexes bind to the plate and can then be quantified. So based on this assay principle, we had already developed a lot of different assays detecting antibodies against a variety of different viral diseases. So we know that our assay is capable of generating assays with a very high sensitivity, is compatible also with the use of high serum concentrations, that we can improve specificity by certain technical means, and we have an easy performance of this assay because there are not so many incubation steps. When we developed this assay, we developed 2 different variants, and the main difference between those 2 variants is the concentration of the serum we use. In this assay variant 1, we use a 1 in 100 final dilution of the patient serum, and here we use a 1 in 2 dilution of the patient serum. And as an antigen, we use the nucleoprotein of the SARS-CoV-2 virus, but in a truncated form, only harboring the C-terminal dimerization domain. When we then analyzed our patient sera from these 35 patients, the longitudinal sera, so the very same serum set that you saw on the first picture, then we saw that if we applied assay variant 1, so with a higher dilution of the serum, we could nicely detect antibodies here in the early phase of the reconvalescence of the patient, but then later on, at time points around 6 months post-onset of symptoms, the signal was already not detectable in the majority of the patients. This was different when we used the very same assay, but with a different serum concentration. Here you see that we are still able to detect antibodies against the nucleoprotein in most of the patients still after 1 year post onset of symptoms. So as you see here, even if this very same antigen is used, the very same assay platform, then the assay conditions strongly influence how long you can really detect those antibodies in the patient's serum. And of course, this also depends on the type of the antigen, so if you're a spike protein or the nucleoprotein, but also for spike-based assays, we recognize that different assay types, for example, the Urimmune IgG ELISA and the Untitled Antibody ELISA, applying different assay protocols, really differ very much in the sensitivity and in the capacity of the ELISAs to detect antibodies up to 1 year post-infection. So as you see, assays are really, generate very different results, even if the same antigen is applied. So what is very helpful in this context, at least to control this a little bit, is the availability of WHO international standard reagents like this plasma here that can be obtained from NIBSC or from WHO. And this is a standard reference sample, and if you analyze this serum with your assay, then at least you have a reference to compare to other assays how the serum performs here. And here you can see an example, different dilutions of this reference serum here, measured with our different assay variants 1 and assay variant 2. And there you see a high sensitivity difference between the two assay variants. If you are in the linear range, you can even normalize to these units here. But even if you are not measuring in the linear range, at least the use of that standard serum can help to get an idea how sensitive or insensitive your assay is. The next problem we encountered is assay specificity. Because at the very beginning, when we did not have our own in-house ELISA, we tested several commercially available ELISAs for assay specificity by using using serum samples that had been collected before the SARS-CoV-2 pandemic. So all these serum samples basically should not harbor any SARS-CoV-2 specific IgG antibodies. And we assayed those samples with different commercial assays, either employing the nucleocapsid protein as an antigen or employing the spike protein. And these are Urimmune assays and the microgen assay that are commercially available in Germany. And as you see here, as long as African sera here originating from Ghana or Nigeria, so to remember you, these are all serum samples that had been collected before the pandemic, so there should not be any antibodies, and as you see here, a lot of false positives are generated here in these assays by the commercial assays. And this is an observation that not only was made by us, but also other groups. We observed this here for Ghana, for Nigeria, but other groups described similar phenomenon, for example, for samples from Benin. So assay specificity depends very much on the origin of the samples, so the immunological background of the proband that you are analyzing there. So if you do seroprevalence studies somewhere abroad, for example in Africa, always the specificity has to reassessed exactly for that target population, just to be sure about the level of false positives that can be expected. And because we recognize this problem, we optimized our own in-house assay with respect to choose an antigen fragment, to the antigen labeling that we used, and to the assay protocol, so that we ended up with a very high specificity, also here in our target regions in Ghana and in Nigeria, where we also want to use these assays to do seroprevalence studies. And here again you see the numbers of the sera that were applied. So we tested around 600 sera from African origin. Another problem that we also encountered when we applied our in-house assay based on nucleoprotein was that apparently for detection of breakthrough infections, assays with a very high sensitivity are needed. because basically when you look at vaccinations that are enrolled in Germany, all the licensed vaccine here are spike-based vaccines, so BioNTech, Pfizer, Moderna, AstraZeneca, and also Johnson & Johnson. All of these vaccines only elicit antibodies against the spike protein. So breakthrough infections should be able to be detected serologically, still by detection of antibodies directed against the nucleoprotein. Up to now, we only have one patient, seen one patient here. So this patient got the vaccination here in June in 2021, and then had a PCR-confirmed COVID-19 infection in October 2021 with the Delta variant, which is currently the most common variant seen here in Germany, and the patient only had mild disease symptoms. Then we sampled the patients at the different days post-onset of the symptoms, or days post-positive PCR. And as you see here, with our less sensitive assay variant employing the nucleoprotein, we don't see any seroconversion here, but we can nicely detect seroconversion, but only at a pretty late time point post-infection with our very sensitive assay. And of course, this is only anecdotal knowledge because it's only one patient. But when you look in the literature, you see also that there are already other groups made similar observations. So, for example, here in that publication, 23 PCR-confirmed breakthrough infections were analyzed, and in 74% of them, even at pretty late time points post-infection, the anti-N assay generated a negative result. So this should be, I think this is a solvable problem, but this has to be taken into account if NCP-based assays are to be used to detect breakthrough infections by serological means, because they're apparently really sensitive assays are needed to still detect those antibodies. Just to sum that up, due to antibody waning, it's very important to know the time correlation between COVID-19 incidence and sampling when you are doing seroprevalence studies, but because only then you can really take into account the assay sensitivity and basically correct the results that you have obtained there according to the assay sensitivity. So the assays that have to be used for seroprevalence studies have to be very well characterized, and also with respect to the waning antibody response and until what time point antibodies are still detectable with exactly that assay. To compare the results of different assay types, standard reagents can help very much to interpret the results. And although the assay specificity for the commercial assays that at least we analyzed were pretty good for German and for European samples in general, this may not be the case when other destinations or other locations are investigated with seroprevalence studies. So always this assay specificity has to be verified for the target population. And then finally, the breakthrough infections after vaccination. The spike-based vaccines can be diagnosed by anti-NCP serology, but apparently, there are more and more reports showing that the responses may be much lower in those patients than in completely unvaccinated and then infected patients. So finally, I want to thank all the co-workers who were involved in these studies here at the BNITM in Hamburg, and also in the different countries we are working together. And of course, last not least, all the patients and the blood donors who agreed to collaborate in those studies. Thank you.
Спасибо, Кристина.
Thank you, Christine. And the next presentation is by Letushev and Dananiev, the laboratory diagnostics of COVID-19 in the Russian Federation. You have 15 minutes. Good afternoon, dear colleagues. In my presentation, I would like to draw some conclusions of laboratory diagnostics in the Russian Federation. And in the second part, I would like to emphasize the importance of laboratory diagnostics at the time of the COVID-19 pandemic. I would like to start with the documents, with the normative documents, and to say that the issues of laboratory diagnostics are in the focus of attention of the government of the Russian Federation. Not a single coordination session goes without detailed examination of testing of the population in different parts of the country. And the president of the Russian Federation, Putin, approved a list of tasks for the government of the Russian Federation. This happened on the 20th of October. And point number 2 is the increase of the number of testings in Russia with the use of the express tests. At the level of the government, we have respective resolutions. There is the resolution concerning the distribution of the systems, and Rospotrebnadzor is responsible for that and carries out the surveillance over the proper use of the assays. And all the organizations that carry out the laboratory diagnostics. And the second one is 4.2, which defines the mechanisms of the notification of the individuals about the results of testing with the use of the Gosuslugi portal. Now, as for the dynamics, we should mention that the work on the diagnostics, the laboratory diagnostics, required the optimization of the work of the laboratories. When the assays began to be made, manufactured, and the laboratories began to be equipped with those, step by step, it was decided to involve all the laboratories of the Russian Federation into this work. As you know, at the end of January, beginning of February, the work in 2022 was carried out just by one laboratory, Vector. But by mid-February, 15 scientific institutions of Rospotrebnadzor got involved, and by that time, no other laboratories were involved in this work. By mid-February, step by step, with the accumulation of experience, 84 centers of hygiene and epidemiology in all the entities of the Russian Federation began involved, and 14 research institutes which had worked in the beginning of February began to verify the results. And by mid-March, in compliance with the resolution of the chief sanitary doctor, the laboratory diagnostics began to be carried out in compliance with a specific document allowing the laboratories to carry out the work with the third and fourth group of pathogenicity. And by the end of March the same year, the state and private medical institutions began to be involved, and the federal center opened the register of these laboratories. And as you can see, there are 1,200 laboratories which are officially involved in the diagnostics of the coronavirus infection in the Russian Federation. Well, of course, mass testing is impossible without proper training of the staff carried out by Rospotrebnadzor, and programs for vocational education and training were prepared on environmental safety, epidemiological safety, and different molecular research and studies. And over 1,000 staff members of the laboratories involved in the diagnostics of the coronavirus infection have gone through this training. Apart from the expansion of the number of laboratories, the training for the staff, we can see that the assays are being manufactured now in much larger numbers. In the Russian Federation, there was only one assay produced by Vector in 2022. Today, according to Roszdravnadzor, there are about 500 medical instruments are being manufactured for testing. These are not only assays, but also different instruments that are needed for the carrying out of testing. So, as a result of the work carried out, the capacity of the laboratory is increased. Now, if we take 2020, February, then there were just 220 laboratories, and 1 month they could carry out just about 400 tests. Today, by the end of the previous month, there are about 4 million laboratory tests carried out. And so we can see an enormous increase, 33 times. And we should also mention that while previously the laboratory diagnostics was done initially by the laboratories organizations of Rospotrebnadzor, nowadays the medical organizations, both public and private, are deeply involved in that. Now, of no less significance is the coverage of the population with testing. 100,000 people. These indicators were introduced and the recommendations were introduced following the recommendations of the chief sanitary doctor. So the minimal coverage was defined, first 70% and then 90%, then over a long period of time, 110 per 100,000 people. In November 2020, when all the laboratory facilities were ready, this indicator was increased to 200 per 100,000 people. And then when it became obvious that all the laboratories were able to reach that level, 200, this reference level was increased to 300. So the average coverage of the population with laboratory tests was 378 per 100,000 people. And we say all the regions are quite different in this respect. So by the end of the 48th week in Lipetsk region, for example, it was just 172. But at the same time, in Yamalo-Donetsky region, this indicator of coverage has not been lower than 900 per 100,000 people for quite a while. So apart from the increase of the number of tests, it's also important to make sure that the results are obtained timely and as early as possible. And today, Anna Popova spoke about this. So with the purpose of following the resolution of 451, on the basis of the Institute of Epidemiology, a system of the aggregation of laboratory tests was created. This system, solar system, makes it possible to carry out the work. So within 60 minutes, we're able to get the results of the testing on our mobiles, e-mail, through applications. And today, over 400,000 results are transmitted like that for laboratories. This is about 80% of all the tests carried out per day. We should also focus on the dynamics of these indicators. Over the past 5 months, they have changed, and this is because of the demand growing from the population. When with certain constraints, it is possible to visit different events, and this Congress, for example, only with a negative PCR test. So the demand increased, and increased many times over. And over the past 5 months, the number of these results increased from 26% to 80% from all the tests there. The ones that are transmitted, all the entities of the Russian Federation have at least one laboratory that is connected to the system and provides the transfer of the laboratory results. And so the work carried out over the past 2 years, as mentioned at the plenary session, the Russian Federation, in the absolute numbers, is among the 5 leaders in the world. And as for the 100,000 people, we're in the group of 10. Among the countries where the population is over 10 million. Now, we should also mention correlation here, because the morbidity, and here you can find here in the regions with the maximal coverage, and the coverage for 100,000 people. Now, the correlation. Now, when we examine the morbidity and the correlation of tests and morbidity, we were able to find the Pearson correlation factor. We can see here the sampling for 5 weeks and 5,400 observations, and we can see here this correlation between the morbidity and the testing, and the correlation between the positive tests and the morbidity. Now, of course, it is logical, because the morbidity in most cases is formed on the basis of the laboratory-proven cases. And when assessing the data and these correlations, we were able to calculate the determination factor. You can see the dynamics of morbidity in the Russian Federation here, which is linear and correlates with the coverage of the population with tests. Less well-manifested correlation can be found in the federal districts, but statistically important still. And in the context of individual municipalities, the numbers here are somewhat distorted, because there are certain regions I mentioned with extremely high coverage, and so they distort the logic field. Yamalo-Nenetsky area and Hunter-Manseyski district. We can see that with the coverage of the population, the morbidity indicators grow, and vice versa. Now, a similar assessment was carried out here of the laboratories tests and morbidity. As I said, we can see there is linear correlation here, because in the official statistics you will find mostly the cases with laboratory-proven results. At the next stage, correlation between the coverage and the morbidity. We classified all the entities of the Russian Federation by 3 groups, and we can find here the normalized numbers, and we can see quite clear that in the regions with high morbidity, laboratories work very well, and there is high coverage of the population with testing. That's why there is quite a lot of positive results. And the districts and the regions with the laboratory work not that well organized and with lower coverage of the population, the morbidity is lower. You can see the difference of these 3 groups by all the indicators. So we can say once again that between the 3 indicators, there is an interrelationship. And at the final stage for the assessment of the correlation between these indicators, a mathematical model was built with the linear regression approach. And this model allows us to explain the variability of the areas. Actually, 40% of the variability explained by the model. 60%, these are other reasons that are not considered here, but which play a certain role in the morbidity officially registered. With the help of this model, we calculated the expected morbidity with the fixed coverage 300 per 100,000 population. In the upper part of the slide, you can see groups of the regions with actually high morbidity. In these regions, as we said, there is a high coverage, higher than 300. If it is regulated and we go down to 300 for them, then we will see the decrease 1.5 times. And there is a very different situation in the regions represented in the right-hand side. Because the coverage there, if the coverage is much lower, about 150 per 100,000, if we increase it to 300, then the morbidity will increase 1.5 times. Thus, we can assume that in these regions, the low morbidity is associated with the low coverage. This morbidity does not really reflect the intensity of the epidemic process. It is just associated with lower number of tests. Now, I think I have said everything, and I'm just going to say that the extensive coverage of the population with testing, particularly at the time of the pandemic, at the time when we have symptomatic patients, is very important, and this indicator is very important when we build mathematical models for the forecasting of morbidity. Thank you. Thank you, Alexander Nikolaevich. And the next presentation is Svetlana Yetsishina from the Central Institute of Epidemiology, specifics of the diagnostics of COVID-19 through the methods of the amplification of nucleic acids. Yes, 15 minutes. As far as I know, it's a recorded presentation. Yes, good afternoon, dear colleagues. Ой-ой-ой, плохо слышно. Now, I speak on behalf of the team of authors. We will speak about the specifics of the diagnostics. With this disease, we came across at the end of 2019. Genetic studies, it is demonstrated that the activator is not quite clear, but there were transmission pathways that were defined. So when at present we are at the height of the pandemic. So what do we know about this activator? The reservoir were bats. And as for the transmission routes, that's contact, that's fecal and oral, and airborne. Now, contact is through contaminated surfaces and dirty hands and virus that ends up on the mucus. And then there is an intermediary host through which people were infected, and this is not known yet how it worked. So the events developed in such a way that the virus found its place with humans as well as other coronaviruses. Here you can see different types of coronaviruses that circulate in the world and that became known since the 1960s. These four viruses are quite well known. Their circulation among people is mostly typical for spring and autumn, and they lead to respiratory diseases, mostly affecting children. Coronaviruses might cause flu-like diseases. Starting with the year 2003, it became known that coronaviruses can overcome the species barrier. And so in 2013, another episode of the overcoming by the coronaviruses of the interspecies barrier occurred. We're talking here about the Mediterranean respiratory syndrome, and MERS coronavirus led to a serious outbreak in South Korea and spread mostly as inter-hospital virus, so with a high level of mortality. In 2019, at the end of that year, we came across another virus, SARS-CoV-2, and there were again cases of interclinical transmission. The main methods of laboratory diagnostics, etiological diagnostics of COVID-19, are diagnostic tests for SARS-CoV-2. And antigens are very important here, and antigen detection, which are coding the genome. And specific antibodies are being detected. If we look at the dynamics of the process of infection, then the probability of getting a positive result is very close to the manifestation of the symptoms of the disease. And even prior to the manifestation of these symptoms, it is possible to detect the activator through the laboratory diagnostics. With the methods that make it possible to find antigens. And these methods work. In almost 20% of cases, the infection occurs, and then the patients get better, and in the process, the concentration of virus goes down, and it is less possible to detect the positive result. It's less possible to get the positive response. On the second week of the disease, the antibodies appear, and IgG, and they can circulate over the next 4 months. When the infection goes through the gastrointestinal tract, then the positive results of testing can be longer than when we take the respiratory form. And, viruses are difficult to diagnose and to find after a certain period of time. So that means we have to rely on the antigens, we have to rely on spit. And of course, the laboratory methods at the bed of the patient are important. The quick tests, the express tests, and these are tests which make it possible to work with RNA and to detect the antigens on the surface of the virus particles. But the express tests do not possess a high sensitivity level, and they're not possessed of high specificity level. And so the definition of the antigen by sensitivity is lower than in case of the molecular work and diagnostics. So here you will find the average data for sensitivity and specificity. In the Russian Federation, there are temporary recommendations for the prevention, diagnostics, and treatment of coronavirus, updated regularly on the basis of the information and knowledge accumulated. And we can see that the express tests for antigens can be used. And if there are positive results, then it's a final positive result. But if there is a suspected infection, a suspicious case, then the antigen and the antigen tests give a negative result, then it is necessary to go further. And these express tests are feasible when in the population we get positive results in over 5% of cases. This is important to reduce the number of false positive results. If pneumonia develops, then along with the biological material, swabs and from the throat, along with that material, if the result is negative, then it is recommended to examine the respiratory tracts then it is necessary probably even to have the biopsy of the lung in order to understand how severe the damage is. In the register of the medical items in the Russian Federation, there are 78 names, titles rather, registered diagnostic kits. And out of them, 14 are isothermal, on the basis of isothermal amplification, LAMP. That is the international abbreviation. Among the registered in Russia reagents for the amplification method, there are used one-step PCR tests, LAMP, separate reactions with reverse transcription, and during the clinical and laboratory tests with the definition of specificity on the basis of computer data or with the application of laboratory studies proving the specificity of the manifestation of this or that coronavirus and the absence of criss-cross reactions with epidemic coronaviruses. So these tests are important. So these tests are most reliable and they prove the level of specificity. Then there are different kits, there are different assemblies of samples which make it possible to control LAMP. And there are also samples that let us know that all the stages of the study have been correctly carried out. Now, the analytic sensitivity was carried out with the use of different methods, with RNA on the basis of reverse transcription, and with the effective extraction methods. When the structures which were used for the assessment of sensitivity were to contain fragments of the coronavirus or have a certain concentration of those. So there can be one target gene or several target genes with a variation. So the results, as a result, are obtained faster than during a standard reaction. And the assay here uses different modifications. As for the time needed for each cycle, this, of course, has an impact on the overall time. And there are recommendations concerning a wide range of biomaterial. The control samples, these are extracts, and they make it possible to control the sensitivity of the analysis in the laboratory itself. The laboratory can assess the compliance of the theoretical possibilities with the practical results, with what is actually obtained and the course of experiments in the laboratory. The internal control and assessment is very important in order to get a more detailed information. It can be carried out at the stage of amplification, for example. And as for the structure, this can be RNA, molecular, which makes it possible to control the correctness of the reverse transcription, and it can be a sort of a cloned exogenic control, and then the methods of control are different, but they make it possible to control the steps and stages of the work. And more reliable results are obtained with the test and the assay that have a larger set of control samples. Dear colleagues, there were several minutes to come, but we decided to make a compilation in order to be within the time framework. We were able to listen to the most important issues next. Dear colleagues, once again, we are to end the session at 5:30, and we have 5 speakers. Okay, so if it is possible for you to make your presentation a bit shorter than planned, then it's going to be good for all of us. Yes. Next, Vadim Petrov, Epidemiological Institute, development of reagents for the reveal of SARS-CoV-2 through the LAMP methods. So my topic, how to reveal coronavirus using the set of reagents developed by us. The pandemic shows that, of course, the molecular diagnostics develops, and the number of tests is increasing very fast. If we take all the other tests and all the way, we can say that coronavirus is far ahead. If we look at the assortment of different projects and products that our institute has been manufacturing, then coronavirus there in the list is just one. But it takes it takes much more space diagnostics-wise. If you look at the methods,
If you look at the methods of diagnostic of COVID-19, of course, the majority of those methods are PCR. However, there are a set of isothermic methods, such as amplifications of the chain, amplification of the ring, other types of amplifications, recombinant amplification, and the so-called loop isothermic amplification, or LAMP in English. So we actually chose this particular option because actually it requires fewer enzymes in order to be implemented. On the other hand, it has a complicated design of primers that represents some restrictions of this method. Of course, it is luckily for the coronavirus, we could overcome those restrictions. We need to choose a conservative area and to choose our nucleotides. Isothermic amplification, loop-mediated as isothermal amplification, requires the temperature of 60 to 65 degrees Celsius. It only requires one enzyme with so-called BST polymerase, which replaces the chains during the amplification and which forms loop-shaped structures. That's where the name of it comes from. So this method allows to diagnose different agents within 30 to 40 minutes, depending on the agent. So what were our objectives when we were developing this set of reactant? We needed speed because mass testing was required from us. Then we needed not to lose diagnostic characteristics, and also we needed a low budget version. The genome of coronavirus contains some target genes that were our candidates, such as ORF1AB, then gene N, gene E, and gene S. Once we made our choice, we opted for ORF1AB because it gave the best results and it had the fastest signal and the best conductivity. Gene S was a bad area because nucleotides were hard to be selected for diagnosis, and variability was there in this region. Our next objective, and the most important one, was to obtain our own enzyme, because we had an internal production of all the components. So it was essential for us to have our own BST polymerase enzyme. So we already had a structure, and then we needed to launch the production in industrial volumes. So we compared that with polymerase produced by NEB. NEB US is one of the leaders of the production of this kind of polymerases in the world. So our polymerase on certain aspect was even better than that by NEB US. So we could localize a key enzyme in our lab, and then what we needed, then as a result, we obtained a kit of reactants. What else did we have as parameters? We needed to speed up the whole process, so we needed to minimize the number of components in order to simplify the reaction itself. We needed also to decrease the size of the testing kit in order to make transportation easier. And also this has an impact on the cost of the analysis. So as the result of our development, we came up with a testing kit which only contained 5 tubes with 4 components. 2 of them are required for making the preparation, and then 2 more serve as controls. The reaction speed, the duration of the reaction was of 30 minutes, and most of positive results could be obtained within 15 to 20 minutes. If we look now at what the preparation of the mixture looks like, at the first stage we mix 2 components, in the proportion of 1:1, and then this mixture is additionally mixed with the nucleic acid that we isolate. So only 2 stages are needed, nothing very complicated. Next characteristics of our testing kit is that it allows us to isolate RNA in the concentration of 10⁻³. As a reactant, we use magnosorb, which is the most widely used reactant kit. We adapted our kit to the amplificators that are largely used in the Russian labs, to what the labs usually have, such as Rotogene, SF, CFX96, and others that were shown on the slide. And once again, let me reiterate the analytical characteristics. The speed of the reaction speed is 30 minutes, and our colleagues from another research group confirmed that in the independent way while comparing our method with the with the traditional PCR. So it was shown that most of positive results could be obtained in less than 20 minutes. 20 minutes is the maximum. As a conclusion, let me say that we have developed and implemented a kit of reactants. We registered this kit. We have a registration certificate. This reactant kit is now on the list of the kits that is provided to the labs according to the decree of the Russian government in order to ensure accurate and fast diagnostic. And what is even more important is that this kit is developed within our institute, and now it serves as a basis for future developments of other reactant kits on the using this technology. So our future prospects is to develop other type of reactant kits for influenza type A, type B, for the detection of coronavirus using 2 targets, because today it was said several times that the virus is quite variable. So in order to, just in case, we are also developing a kit using 2 targets. So far we don't need it, including owing to the database of VGA-RAS. However, we are closely monitoring this process. We are monitoring the emergence of new mutations, and if case we need it, we already have some preliminary design of a kit with 2 targets. We are also developing a kit for mucotic infections that have already been covered today. That's also crucial to have one like that. So in general, this solution, this technical solution, could be referred to as a basic platform-oriented solution. So to conclude, I would like to thank all the researchers of our lab and different entities of our institute. I would like also to thank those employed by the group of gene engineering and biotechnologies and of PCR laboratories, as well as of the laboratory of new diagnostic methods. Thank you very much for your attention.
10 минут. Благодарю. Следующее сообщение.
And the next presentation is on the experience of the development and implementation of the portable diagnostic systems for the pathogen manifestation through the method of isothermal amplification. Yes, you have a bit less than 15 minutes, please. No, thank you for the chance to speak. My presentation is dedicated to the development of an analyzer. Right away, we will look at the apparatus for genetic tests and analysis. In most of the laboratories, you will find the tablet-like instrument to work with the micro glass tubes for testing. This equipment is justified when we take a lot of samples. But the experience of coronavirus epoch, the experience of work of the diagnostic center of our center shows that it is necessary to have express diagnostics with a limited number of samples. So we did not want to interfere into the work of large amplifiers. So it is necessary to have the portable ones. It is necessary for the laboratories to have those mobile, these portable kits. So our purpose was to satisfy this particular need. And as a result, we obtained an analyzer, Isoscreen 8, which makes it possible to carry out the testing through isothermal amplification and carries out exactly the same work as the large devices and makes it possible to detect the pathogen. So the key specific feature here is loop isothermal amplification. The previous speaker described it in detail, so I'm not going to go back to the story, but very briefly still. The main character is BST polymerase, which makes it possible to carry out the work at constant temperature. Optimal temperature for the enzyme is 60 to 65 degrees, and the loop-like structures, amplicon, within it. So the amplification should be always at a constant temperature, which makes it easier to work with the equipment and also makes it possible to speed up the amplification because the cycles are not there any longer and the reaction is continuous, and thus the time needed for the analysis grows shorter. Now, how does it work? Using the example of coronavirus, the system was registered with a set of reagents, which are called ESAMP, produced by Litech company. So, well, of course, first of all, the swabs are made. Then the samples are taken to the transport buffer. Then from there to a special reservoir. The analyzer makes it possible to use the method of thermal coagulation. And there is a glass tube heated to 95 degrees over 10 minutes. And then the next stage is the amplification stage, for which we need to carry out the reverse transcription, so RNA into DNA. And this happens with the help of a special enzyme at the 50-degree temperature. And then after that, amplification in real time, and polymerase collects amplicones, loop amplicones, and then the larger is the number of those amplicones, the better is the signal from the sample from the tube. And the optical system registers that. The amplification takes about 12 minutes, 65 degrees centigrade. We should also mention here that in principle the analyzer is universal. It can work with other test systems, with other assays and reagents, and it is possible to change the time intervals. For example, if there are other methods used for different of these stages, we can actually exclude the procedure of the extraction. Now, let's have a look at the system itself, at what the device looks like. It's a compact size, modest size instrument. There is a screen, as you can see. On the upper panel, you can see the thermostat, and there are glass tubes there for the carrying out of the work. And on the slide, you can see the cross-cutting of that device, and you can see that the heating elements here repeat the shape of the glass tubes. And then there are standard PCR glass tubes, which are placed into the modules of amplification and detection. And there are two of them. They are located in the side panels of the analyzer. And in these modules, the procedure of reverse transcription occurs, and the amplification happens there as well. Now, in the lower part of this slide, you can see the schematic representation of the instrument. You can see that there are no moving components, so the system is quite stable. There's nothing there to break and to be broken. There are 8 irrespective detectors working there. They're all tuned to one and the same filtering range of 520, 560 nanometers. This particular range is very often referred to as FAM. This spectral range is used in our analyzer. There are only 2 independent heating coils heating the glass tubes. There are 8 of them altogether. As a result, we get the temperature dependence occurs. Briefly, once again, the main technical characteristics of the analyzer. It's compact, it's mobile, sensitive. It weighs about 1 kilo. Can work on battery or can work through the power supplied by the tablet or the mobile phone. Two independent heating elements for amplification. Where can is a screen 8 be used? And why we designed that particular analyzer? First of all, we need to say that for reliable analysis, it is often necessary to carry out other types of controlling measurements. And so, in order to carry out the analysis of one sample, at least 3, sometimes 4 glass tubes are needed. That's why this particular structure is important. 2 amplification models, 4 by 4. So the instrument can work with 2 samples at the same time with the reliable final results. For the set of reagents, we suggested a scheme that makes it possible to simplify for the operator the procedure of the analyzing as such. The reagents are initially in the glass tubes, placed in the glass tubes, and it is enough to place the samples in a proper order into the analyzer, and the time of the analysis is less than 30 minutes altogether. Moreover, the software of the analyzer makes it possible to carry out the automatic analysis. So instead of the graphs, you can see the diagram, the infograms of the results. Yes, we're stepping away from the use of the tablets, from the streamed processing of samples, and we hope very much that this scheme is going to be useful for the express analysis, for the bedside medicine, for the mobile laboratories. At what stage are we now? It took us about half a year to develop the instrument. It has gone through technical testing. The clinical testing and trials are ahead. And the documents have been submitted to the patent agency. And we have made a small set of instruments. Some of them are in the hands of our colleagues at the Russian Scientific and Research Center, the Meragenomics company, the tech company, in order to ensure the testing of the instrument under conditions of real life, in order to be able to catch, so to speak, all the new diseases. What are the prospects? Where should we go from here? The global trends indicate that molecular genetic tests are feasible in the automatic regimen, in the disposable cartridges and microfluid systems for micropipes, microcapillars, microchannels, microreservoirs and pumps, which exclude manual work of the operators in the process of carrying out the analysis. Everything is carried out by the robots, and we're moving towards that indeed. At our center, there is a laboratory at which the complete cycle of photolithography is being developed, and we can create the prototypes of these systems and cartridges there. Speaking about the experience of work, we can say that in spite of the visible simplicity of the analyzer, it looks quite simple. But in reality, there's quite a lot of equipment needed for its manufacturing, and design bureau is needed for that, not just a laboratory. And, well, of course, a team is needed. Molecular biologists and engineers, designers, physicists, programmers, engineers. For the effective work of this team, good management is paramount. So that's all I wanted to speak about the development of the analyzer. Thank you. Thank you, Kirill Alexandrovich. The next presentation, Organization of Mass PCR Testing in a Large Centralized Laboratory by Andrei Komarov, the Diagnostic Center of Laboratory Studies in Moscow. I'll try to be very brief. I wanted, first of all, to thank the organizers of the forum, Anna Popova, and to say a few words about. We're a large specialized organization. We carry out just laboratory studies for Moscow, and I'm the director of this center. Now, for us, in March, was set the goal of mass testing. At that moment, we had several problems facing. First of all, shortage of space and human resource equipment, and shortage of everything. Systems and assays and Vector, for example, at that time, worked through their own stages of work with reverse transcription, and it took quite a number of hours to go through that process. And Rospotrebnadzor physically could not cope with the growing number of tests. In Moscow, at that time, the capacity was 10,000 PCR tests per day. 6,000 of them were routine tests, and 4,000 were free. And according to assessments, which were corrected later, we needed 40,000 per day. So the increase was tenfold. So that is why it was decided to organize centralized laboratories, because of the concentration of human resource and equipment and resources. With centralized laboratory, it was easier to be more flexible, to provide better supply systems. We were reporting to the mayor on all the details of work of the laboratories at that time. And within a very short period of time, as a result, we were able to organize mass testing. By May this year, Moscow was among the leading cities in this sense, number of tests per capita. The budget laboratories were carrying out about 40,000 tests, and then commercial laboratories began to work. And so let me speak about our center. The major principle of the selection of technologies. These technologies need to be scalable, standardized, should be inexpensive, reproducible, and they must be sensitive and specific. So when we selected the technologies for COVID diagnostics, then of course that was etiological diagnostics and the investigation of the immune response. So PCR tests, antigen viruses, and genetic studies, NGS and fragment sequencing. And respectively, by all the parameters, the PCR diagnostics was right there. Antigen was not that precise at the time. As for the genetic investigations, that was too expensive, and there was no capacity for that. And response, immune response, antibodies count, IFA methods, and immunochromatographic methods of antibody detection. And 100 devices were bought, and reagents were bought for 14 million couples, and we're still using them. So specifically in our center, a special site was designed for HL and PCR within just a couple of weeks, with ventilation and everything else, and power supply. Thing like that in the past, and so 65,000 per day. That's how many we can do. At the same time, we integrated all the information systems, EMIAS, the Moscow system, LIS, and the system of Rospotrebnadzor, and the data exchange was organized between different entities and agencies. All the laboratories began to work in the conveyor style. The functional zones were created. Without that, it was impossible to increase the productivity level. So we had a check-in zone with the sorting of samples. And this is the conveyor work. And of course, the work was 7/24, because with the deficit of equipment and human resource, that was the only type of work, the only method of work that was possible. Anastasia immediately set a goal for us. All the results were to be presented within 24 hours, and this was strictly followed. The mayor's office monitored the situation. The routing, the samples, and the issuing of the results. All this was controlled, and for the this period of time has been cut to 12 hours, and within our laboratory was a special sector where everyone responsible for that was working. That's how we work. the different functionalities are represented here. The area where we work with the applications and the courier zone and so on and so forth. And here you can see that's where the couriers are. To the right is the window for the PCR tests to be handed over. The main functional blocks, if we take PCR, here is the zone of the detection and the amplification zone. All the work carried out here was carried out on the basis of the integrated product, and then once all the work was done in LIS, then again everything went back to the MES system. Look at the immunochemical analyzers. We have 9 of those with very high productivity level, and we're happy with that. That's Andrei company. Although attitude was different at different periods of time. That's about about 200 tests per hour per machine. Here they are actually. Immunochemical devices. Now the dynamics of the productivity. If we take April, May, then the curves were almost identical. In the summertime, we were able to get this technological gap between the 2 lines. And we were at the limit of our capacity at that time. Any sort of a failure could lead to non-compliance with the promises. And the laboratories were equipped in the following way. There were amplifiers and all the other instruments were coming one after another. Then the systems were to be adapted properly. Tuned, and so the optimal protocols were introduced, and all this has an impact on the productivity level. Now, if we look at the graph, these are real graphs of productivity and research carried out. We can see the waves of COVID here, respectively, and the productivity delta. You can see how well we can scale our work. With exponential graphs, gentle graphs, that was typical of the other approaches. But in immunochemical analysis, that looked like that, with a sharp increase, with the possibility to work with any volume of antibodies. Now, and in May, we started a project on express testing for mobile teams in the airports. So isometric colorimetric reaction was part of this work. So as a result, we were able to provide the result within 1 hour in any language with QR codes as needed. And the work itself was carried out at high quality level, but within 25 minutes, so with colors that work quite well. The sensitivity is lower than the standard PCR system. The manufacturers quite agree with that, but for mass screening, it's all right, it's acceptable, particularly since we can set it up in any place, in any location. Then, as an experiment, we began to carry out the semi-genomic sequencing of virus. And since August, we carried out over 3,000 of sequences. And so 2 luminars are completely loaded twice a week. That's how we work now. Now, if we look at the sequencing, what is being sequenced? 100% delta. One virus was Russian Finnish, just once. And so it's delta, always delta. And if we take the Russian variant of delta 1 to 2, we 83% of 1 to 2. The future. Now, if we look at the COVID epidemic, then most of the clinicists, with all other equal conditions, say that the disease there interacts with viruses and the system, the human body. So we study the genetics of the body and give forecasts on how the system is going to overcome it, but there are not that many researchers dedicated to that. And there are PCR multiplex methods with bioinformation. That's what we're working on, the target sequencing of receptors and complete exome sequencing in combination with genetic chips. And then a retrospective search within the human genome of the factors that affect the course of the disease. The population seems to be the same, but all people suffer differently from one and the same disease, and these are the factors that need to be studied. Our conclusions. First of all, centralization. Without this centralization, it is impossible to carry out mass screening and mass testing for COVID-19. The pandemic required that, of course, and the capacities in the centralized laboratories quickly got scaled and could be used effectively. And, well, of course, the growth of the automated systems in the assessment of immune chemistry can get scaled much faster than PCR. PCR is much more labor-consuming, although it has turned into a conveyor methodology. I can speak for Moscow and for our laboratories, and Moscow is ready for any development, increase of the testing twofold, for example, even 3 times. And in practice, step by step, there are genetic studies of human beings, and as it has been said, future is with LAMP PCR. The vector is towards the bedside diagnostics, and it is dictated by the system of public health, and it's going to be true not only for coronavirus, but for other infections as well. Thank you for your attention. Thank you very much. The next speaker. They represent a laboratory, actually. I suggest that I'm not an organizer initially. No, time is limited, so no more than 12 minutes for each of the remaining presentations. What would you say? Now, we will go beyond the allocated time, and we still have a discussion. So scientific and practical work of the laboratory diagnostics, the experience of the Archimed Clinic. Ladislav Bezrukov. And so we are going to sum up the experience of work of the Archimed laboratory on the organization and carrying out of the laboratory diagnostics of coronavirus infection. A few words about the laboratory. Our laboratory is a private laboratory, multi-profile laboratory, carrying out a lot of laboratory studies. It's a basis for clinical laboratory tests of the new laboratory equipment, and it is also a basis for the training of specialists using the new methods of laboratory diagnosis. The Archimed laboratory can work with the pathogens of the 3rd and 4th group, and carries out the following diagnostics using the following methods: morphological, molecular, biological, serological, and indirect methods of diagnostics. And 3rd of March 2020, the assessment of our capacities was carried out, and a special division was created for the diagnostics of SARS-CoV-2. It is number 3 dangerous coronavirus identified over the past 20 years, and it's the second group of pathogenicity. And our laboratory has all the necessary permits to work with the 3rd and the 4th group, can carry out the diagnostic work with this pathogen only, and we use amplification of nucleic acids without work with the live virus. And we using methods of etiological diagnostics. We first of all detect RNA with the application of the method of the amplification of nucleic acids, and then detection of antigens with the use of immunochromatic methods. The problems of the initial stage of the organization of laboratory diagnostics were as follows. First of all, biosafety. There was the absence of the reagents necessary for PCR testing and also for detection of the nucleic acids. There was no equipment, and there was a sharp deficit of the individual protection means. Inactivation of the virus at the site where the biological material is taken, that is the optimal variant for us. We suggested a transport system on the basis of guanidine with ditreatol and Triton X100 added. It was demonstrated that the virus was completely inactivated. As a result, this was demonstrated in the process of testing together with vector. And RNA of the virus was retained for a long period of time, up to 7 days, and at minus 24 degrees, it was retained for a much longer period of time. And it also became possible to create the laboratory panel for the work with the amplification method. Then it's quite possible that the surface of the glass tubes can be contaminated with the live virus. For that reason, we carried out the disinfection using the 70% ethylene alcohol. Now, we worked with the individuals who did not have symptoms of acute respiratory infection and who were not in contact with the COVID patients. So that was a very important element of safety provisions for us. We, of course, to avoid any sort of contact for these individuals with the potential carriers of the virus. And we used mobile medical modules for the taking of the biological material with the purpose of protecting the majority of those who worked in the clinic. Now, the first diagnostic work was the portable mini laboratory Evotec Mirai Genomics, which was properly registered, and within it there are reagents for the work with RNA, for the reaction of the amplification, for the isothermic reaction, and there is also a set of documents, as well as the amplification, isothermal amplification for the detection of RNA in samples. I'm not going to describe the principles of work. Let me just say that here we use the lysis of the biological material in the inactivating medium. We carried out the comparison of the diagnostic effectiveness of the reagents with the use of amplification as compared to the classical PCR with reverse transcription. And we discovered that there is a high diagnostic efficiency, and the sensitivity was 100%, and the diagnostic specificity was over 90%. And with the acquisition of the registration documents for different kits for the detection of SARS, we carried out the assessment of the efficacy. Now, there were kits, not only the ones that are listed here, but also the ones of Litech. They were all highly effective. So we also looked at the optimal sets for the RNA detection, and it appears that the combination of the kits based on the use of the solid phase extraction for silica gel applied to the magnetic particles and magnetic processes for automatic work reduces the time of the RNA detection to 60 minutes. How can we use the portable laboratory? We did not know the answer to this question. And so together with Rospotrebnadzor, we described certain conditions for the use of such a laboratory. So it can be used within the conditions of a stationary laboratory that has the right to work with the respective groups of pathogens. Also for mobile work on site on the basis of a mobile medical module or within temporary laboratories in field conditions. But with all of that, it should be a part of the clinical diagnostic laboratory of a stationary type. What are the conditions of the use of these portable mini laboratory? The territorial body of Rospotrebnadzor should be always informed about the place and time of testing. It is necessary to organize in a proper way the work with the patient, with the clients coming, and the classical requirements to the PCR laboratory. There are 2 rooms needed in order to avoid collision of the flows of people. And it's necessary to use disinfecting means UF and think about the waste recycling. Now, testing on site. How is that organized? First of all, we can take the biomaterials on the basis of a stationary laboratory. We can organize mobile laboratories, and we can also, in certain cases, open temporary laboratories in the airports, for example, for the testing of the passengers to make sure they do not have the COVID-19 infection. These are the main aspects of our work described here. Not going to describe them all. Inactivation, of course, it is one of the most important methods to ensure safety of those who work in the laboratory. Then it is also necessary to mention that from the moment when WHO declared the pandemic, we began to think about the cutting of the time needed for the processing of the samples. 24 hours initially, but when we started thinking about the round-the-clock working scheme, and well, of course, it's optimal to use the test systems based on isothermal amplification in order to reduce the time needed for the carrying out of the analysis. We have a portal for independent online registration necessary for the organization of the process and in order to prevent unnecessary contacts. And we also work with the respective databases. And what are the results of our work? Over the period of time of the pandemic, the first testing was carried out on the 24th of March 2020. And since then, 4.5 million tests, which is 2% of the total number of tests carried out by all the laboratories of the Russian Federation over that period by the 1st of December this year. Now, for the Moscow organizations, the public order, it's over 100,000 positive tests, 91,000. And now we are testing healthy people without any symptoms, and they are the ones who do not have and who did not have any contact with the corona patients. And the maximum number of tests per shift, 15,000. Average diagnostic work at present, that's 5,500 tests. The method of isothermal amplification makes it possible to reveal RNA within shortest possible time. The application of this method is the most promising method for the development of assays and testing systems for the diagnostics of the agents in order to make sure that the early treatment provides for better outcomes. And for those cases when timely express preventive work prevents the development of a disease, for example, in case of the tick infections. Next, flexible systems for diagnostics and epidemiology. Laboratory Litech, Moscow. I would like to thank the organizing committee for the chance to be here and to speak. Our colleagues have been saying quite a lot that the new infection unexpectedly turned into a part of our life, and at this moment, combating it, the examination of the epidemiological situation, carrying out of epidemiological measures is an inevitable process of all the systems of Rospotrebnadzor. And Rospotrebnadzor, in this case, is taking the leading position in this work and should build hypotheses for the monitoring and diagnostics, and we are to help them with the implementation of these goals. Such practical laboratories as ours can help with that in a practical sense, because we can help and find new compilations, new solutions for well-known processes using our own capacities. One of the main goals for Rospotrebnadzor is daily testing of the population for the virus, and our laboratory can help exactly with that, using minimal capacities, limited space. Let me say that, well, of course, automation helped us with the tackling of these particular issues. Now, it was not that easy for us to arrive at the conclusions, at solutions, and 6,000 or 7,000 tests for us were too many. We were helped because there was a large percentage of positive tests, and as a result, Rospotrebnadzor allowed us to get the status of their laboratory along with 2 commercial laboratories. As I said, it was difficult for us to cope with the load of work. We had just 7 amplifiers at the time, and at that moment, the problems were resolved simply by a large number of boxes for biological protection, where all the manipulations were carried out manually. Plus to that, there was another problem for the time moment. The material was coming in different forms, and this slowed down the process. And epidemiological situation was worsening, and in autumn 2020, the situation required new solutions. And the new solution for us was the acquisition of 2 stations, which it was difficult to incorporate into the process of after all the manual work we had been carrying out. But nonetheless, the monotonous practices made it clear to us that automated solutions were definitely much better. And we worked with different organizations on the delivery of the biomaterial to us in the uniform boxes, so to speak. And so the process was standardized. At the next stage, now the process was still quite a lengthy process, because we used at the stage of the transport of RNA, we started using the tablets, and it became possible to use our stations to the full capacity. Now the transport process accelerated, and then there appeared the new tablet-like amplifiers, and at this moment we are able to work with about 10,000 samples. And most of the staff in the laboratory works in the with the automated systems, and thus we were able to meet the next pandemic wave well prepared. Here you can see the thermal shakers that we were using, and we kept developing, because we know there is no limit to perfection. And we continued testing. We kept looking for the new formats, and for us, the format 384 is the one we're testing now. All the algorithms are exactly the same as the one for format 96, except for amplifiers. Sorry. No, if this method is approved as an effective method, then we will be able to make to work 4 times faster, and we will be able to work with a 4 times larger number of samples, and we will exclude the human factor. And I think that that's important. And the time between the waves, we were able to use the accumulated experience to take part in a project together with the clinicists in order to study the concomitant mucus infections. And Sergei Vasilievich spoke about those. He spoke about the mucus infections, and of course the heavy course of the disease and the artificial ventilation and cytokine storm immunosuppressive therapy, they all increase the risk of bacterial contamination of the bronchial tree and the emergence of the new strains of microorganisms and concomitant infections. And the mucus infections can worsen the course of the treatment and actually can make treatment less effective and can lead to higher mortality rate. Here you can see the data we were able to obtain, and the patients were getting the now we took the swab from the patients, and there were 3 stages of the work. There were seeding and microscopy. Thus, in 37% of cases, we were able to reveal the growth of Candida albicans, and in 2 cases, Candida krusei. And in all samples, we discovered either spores, mycelium, or some other elements of the mucus. So we concluded that early diagnostics is very important in order to exclude the heavy mucus infection with patients with severe COVID. And one final thing I would like to speak about, it is the experience of sequencing, another process of testing and improvement of our work process. As I said, with limited capacities and limited space, we began to work on this new stage, and it's being tested now. So we selected the sequencing method, and the had to work with the solvent method at the laboratory. What is the advantage of the use of this particular type of sequencing? The size of the equipment and the minimal space needed for that. A visual image of the results obtained is quite a good illustration, and it works for each and every sample and for a group of samples. And our experience of practical laboratory makes us conclude that it is possible to use simple and flexible diagnostic methods in order to resolve rather complex and sometimes resolvable problems. And these solutions should never be put aside. It's a possibility to work with them, and it's also a possibility to revise the long-standing standards in compliance with the reality of the time. Thank you. Thank you for your attention. Thank you, Oleg Borisovich. And now, discussion and presentations. Now, 3 minutes. Serological tests and the definition of the status of the new coronavirus. Madam Osanova. Tashkent, Uzbekistan.
Good afternoon, dear participants of the conference. Thank you for this opportunity to present my report. Within the time limits, I'd like to report on the situation in Uzbekistan. Could the organizers please give the presenter a clicker? The COVID-19 pandemic in Uzbekistan is part of the global pandemic of COVID-19 due to a SARS-CoV-2 virus. We know that antibodies against SARS-CoV-2 are usually identified within the first several weeks after infection. they indicate that the person was infected with SARS-CoV-2, independently of the fact whether the infection is severe or asymptomatic. In Uzbekistan, there are different options for test kits to identify SARS-CoV-2 antibodies. They meet all the international requirements for express testing systems. The Agency for Sanitary and Epidemiological Welfare in Uzbekistan conducted a research with the aim to study different options for SARS-CoV-2 vaccine, and also to detect the immunization level in those vaccinated, thanks to comparative analysis and thanks to testing the resulting vaccine. So we conducted a research in more than 1,000 people. 417 were non-vaccinated. All the rest were vaccinated. There were 1,300 women and over 300 men. 51% got the vaccine against SARS-CoV-2, then some AstraZeneca, and all the others received the Sputnik V vaccine made in Russia. The age was comparable. It was the mean age was 38 years. As for IgG and IgM, their rates were also comparable. However, here we identified some divergences between 0 and several thousand units. Therefore, we divided the patients in two groups, under 100 and over 100. So the basic unit was 1 milliliter. And this allowed us to compare different experimental groups according to their antibodies rate and to the severity of the disease they experienced. The analysis showed that those whose IgG titer was between 100 and 1,000, their number was as high as 67%. However, in those vaccinated, independently of the type of vaccine received, the level of IgG was more or less similar and was generally below 100. Was generally between 100 and 1,000, with the mean value of 500. As for the antibody reliant domain, in those non-vaccinated, in 48% of cases, it was between 100 and 1,000 units. And this titer was more frequent among those vaccinated. Further analysis showed that in those non-vaccinated, the required level of antibodies was achieved after 10 days to 1 month. So the speed of immunity development was the highest in those who received Sputnik V. They were forward ahead of all the others of 10 days. So it was a good titer. The titers were high after 1 month. Following 1 month after the vaccination. Here you can see the titers of over 1,000 units. The similar picture was observed for IgG, that was a response to Sputnik V vaccine. So here you can see that the time of immunity development was 18 to 20 days. So as a response to vaccination by AstraZeneca, antibodies were developed within 7 to 10 days. The difference was that the frequency of high antibodies titers was higher after the first dose of vaccination. Here you can see that there was no statistically significant difference between different groups of those vaccinated, and we need to further study the capacities of ZF-UZ vaccine. So our results show that the titer of antibodies after this, the introduction of this vaccine is quite high, and that it's quite comparable with other vaccines. However, the most efficient vaccine was Sputnik V. AstraZeneca took the second place, and the Uzbekistan-based vaccine was also efficient, and the antibodies were developed faster than in those vaccinated was AstraZeneca. So we say that we can say that usually the number of antibodies was as high as 170 milliliters in all those vaccinated. However, we still have a ground to compare different types of vaccine. Those non-vaccinated also developed antibodies within 8 to 10 months after they had the disease. So the number of antibodies developed was sufficient. In 50% of those vaccinated, the rate was higher than 170 units per milliliters. As for the time of the development of antibodies, Sputnik V was the most efficient. So those vaccinated with Sputnik V were ahead of ZF-UZ by 5 to 7 days, and ahead of AstraZeneca by 16 to 20 days. The Uzbekistan-based vaccine was also efficient. As for the time of antibody development, it was ahead of AstraZeneca by 10 days. So the reaction was more or less similar after the first and the second dose of vaccine. However, there were more frequency of higher antibodies rate after 2 doses than after just the first dose, which means that the second dose prevents the elimination of antibodies and contributes to the maintenance of solid immunity. Uzbekistan continues to work on ZF-UZ vac, and we also mean to compare that with Pfizer vaccine. Thank you very much for your attention. Thank you very much. Is Natalia Georgievna here in the room? Are you going to present, because you didn't intend to? You're not on my list. So are you still intending to make your report? Yes, I am. 3 minutes. Thank you very much. Can you please display my slides? Quality of testing kits for diagnosis SARS-CoV-2 during the pandemic in Uzbekistan. Thank you very much. I'm grateful to all the previous presenters. I just have some more to say. We had very similar issues that were common for everyone who is present here. We faced the pandemic with all the problems, such as the lack of equipment, the lack of personnel, and the lack of methodology. The first testing kits that were delivered to the Republic were the test systems produced by Vector. So since January of this year, you have been producing this kit, and we regularly receive them. I mean the test kits for screening purposes. The first positive case was identified using this test kit, and the confirmation was ensured by TIS Morbid test system made in Germany. As the pandemic progressed, our country started receiving other test kits produced in different countries. So the question was which of them were more efficient, because the sensitivity and the specificity of those test kits couldn't be assessed in a short time. Therefore, the question was how to control quality of those test kits that we received. We already had some experience in terms of quality control. We are doing that within our lab. We serve as a reference lab We've been doing that since the early 2000s. We were assisted by a Swiss lab of quality control, which jointly with the Ministry of Health of the Republic of Uzbekistan deployed this laboratory. So we had quite a few control panels for viral hepatitis. So we've accumulated a solid experience over 20 years, and the first samples that we received allowed us to design a new panel to test future cases. So we stored all the positive cases identified through PCR. If we also had clinical data, those came from the cases that were under treatment in the Virology Institute. So we assessed their titers, and that's how we created our own panel to design subsequent testing kits. So you can see that within 2 years, we've tested PCR test kits, express test kits, but also those that were meant to identify either IgM or IgG or both. We also stored the samples of blood serums that we took from positive cases. So we have a whole collection starting from day 1 to reconvalescence period. So we are still storing all those samples, and we have a complete panel today. According to the data received through test system, we can see that the specificity of express tests was close to 100%, whereas specificity didn't reach 100% in any case. As for the kits that were under clinical trials for PCR, there were some interesting cases. So as a basis, we took BGI made in China RT-PCR kit because their sensitivity by the time was 100 copies, the best one at that time. So with positive samples were taken as a basis for all subsequent tests. And this system was served as a control for all the test kits that were delivered to Uzbekistan. We also have our own designed test kits, one coming from the Ministry of Innovation that followed a clinical trial in the Virology Institute. By the end of 2020, we also registered one more kit that was produced by the Academy of Sciences. This one has 2 versions. The LAMP-based version, which is very similar to that presented by Russian colleagues, and colorimetric version. So this year they are registering 2 more testing kits. The first one is Express RT50, and one more. Thank you very much for your attention. That's our news and our achievements in this field.
Thanks.
К огромному нашему сожалению.
It's very unfortunate, but we need to end up. As far as I understand, each of the speakers could say a lot, but time to draw conclusions and to move on following the program. As Anna Popova likes to say, the pandemic is a driver for the development in many different spheres, epidemiology, virology, immunology, and I'm sure that our session today is the indicator of how what the situation was like previously. I mean, we were not ready for that. We all remember March and April 2020, the situation was most difficult with diagnostics, and it was semi-dramatic in 2020. And it was mentioned there were just 4,000 tests. This sanitation system of Moscow did. Just our institute in June was making 7,000 to 8,000 tests. But every one of us has gone through this stage of development in their own way. And today we spoke about rather difficult things, psychologically difficult. For those who worked in the field, we did not have methods for specific protection at that time. I'm talking about individuals. I'm not talking about the test systems and assays. I'm talking about the human resource. Today we speak about the technical aspects. We did not touch upon IT technologies, without which the mass production and the research are impossible. Let's not forget about our people who created this magic. Without them, without specialists who were there in their workplaces, nothing would have happened. And just plastic laboratories would have worked without our people. Today, we have moved already to the highest quality, and this would have never happened without the true professionals who work in the field. Today, we can see that the diagnostic medicine has made huge steps into the future. In the past, the analysis, the blood test and blood examination for fragments was a good achievement. Today, we have the PCR diagnostic is a routine type of diagnostic now. We with lump, we can carry out this work within just 1 hour without any lower quality in the sense of sensitivity and specificity. Today, we go even further. Today, they are asking higher speed of sequencing. And sequencing is the highest level of diagnostic studies. And today, we have accelerated all this work. Today, we can sequence with the preparation of samples within just 2.5 days with the proper result provided. Isn't that magic? I also would like to say that diagnostics of any infection, that is the initial stage, but this is also the most important element, because in any situation we can have the proper diagnosis only if we know what the pathogen is, when we detect the pathogen using the officially accepted methods, and when we can carry out the respective analysis. And today we are at the point of not just diagnostic, the coronavirus, but we can also define the China variant and subline. And all this can be done within 2.5 days. And so I would like to thank all the participants of the session today. I do think that the vectors designed for us from the point of view of the effective and rapid diagnostics are still with us. We are every single time we are amazed at the new methods that actually surpass the old methods greatly. And I would like to wish all of you creativity in your work, success and achievement of the highest goals in the interests of the patients and for the purpose of the prevention of diseases and health of our people in our country and our friends and colleagues all over the world. Thank you for the session. Thank you.